Hi, and welcome to another episode of Tom Kennedy Science. And I'm your host, Dr. Tom Kennedy. Now today, we're going to talk about cellular respiration and mitochondria. I know, it seems like there's all kinds of videos out there on the web about that, and here I am. I've got one of my own as well. Well, there's a reason why there are so many videos of cellular respiration on the web. The reason why is the cellular respiration is incredibly important for biology. I mean, think about this. Cellular respiration is at the heart of our metabolism, right? I mean, linking catabolic reactions with anabolic reactions. And it's how we extract energy from our environment. And not only that, I mean, cellular respiration and the origins of mitochondria it paved the way for multicellular life. And it gave us the two sexes. Sexual reproduction is involved with aging, and it's even involved with apoptosis. So yeah, cellular respiration, incredibly important. I mean, this is central to us. So what exactly is cellular respiration? Well, cellular respiration is this redox reaction, right? We're oxidizing organic molecules in the presence of oxygen and breaking them down to carbon dioxide and water. And what we're doing is we're creating intermediates that can be used for anabolic reactions, hence, the link between catabolic and anabolic reactions. And most importantly, we are extracting energy from the foods we eat to make ATP. And that's important. Now, like a fire that you see here going, that's one of our old campfires. Cellular respiration, redox reaction, but there's a big difference between a fire and cellular respiration, even though they're both redox reactions. This is a spontaneous reaction that's uncontrolled. There's no enzymes going on with it. It's just going to go until it runs out of fuel. Whereas cellular respiration requires, oh my gosh, glycolysis, pyruvate oxidation, Krebs cycle, and then also special structures for the oxidative phosphorylation. So there's like almost 18 chemical steps involved with cellular respiration. There might be even more than that, actually. So yeah, cellular respiration is quite complicated, unlike our fire here, but they are both redox reactions. Hey, and you know another really big redox reaction? Yeah, you ever seen a rocket launch? I grew up in Florida, and even in North Florida, oh my gosh, one time I thought I saw some aliens once, and what I really saw was a space shuttle launching. But I'm in the Panhandle. I had no idea you could see the space shuttle from hundreds of miles away launching. It was so cool, and I did not take this photo. I snagged this one from NASA. Thank you very much. But whenever you watch a large rocket launch into space, they got to go 7.5 miles per second. They're on a very large redox reaction that's highly... Okay, so you guys remember redox reactions, right? So with cellular respiration, our overall chemical reaction, we're going to take glucose. Now, it can be other organic molecules, including lipids, and we're going to break that glucose down into carbon dioxide and water. Carbon and hydrogen, they're getting oxidized. They are going to lose electrons. Basically, what we're doing in these first three stages of cellular respiration is we're going to rip those hydrogens right off of them and take the electrons with them. And we're going to carry them over to power oxidative phosphorylation. Meanwhile, oxygen is going to get reduced as it forms water. So that's the overall thing going on here. And the whole idea of this is to make ATP, adenosine triphosphate. This is a high energy molecule, and it is the energy currency of life. I mean, every single cell on this planet makes ATP and uses it to do work. It's pretty amazing here. So here we go. Let's dive right in. Now, a lot of people talk about the steps or stages of cellular respiration. Some books and some resources, some videos, they've got 3 stages. Some have 3 in a linker. Others have 4 steps. I usually go with 4 steps. And I consider this one called pyruvate oxidation to be an individual step. However, I think it's probably better thought of as a linking reaction between glycolysis and Krebs cycle. Now, since you haven't seen this video yet, you might not know what glycolysis is and the Krebs cycle is. Now, whenever I teach this in class and you have to come and take a test on cellular respiration, I always say learn the big picture first. Memorize that equation of cellular respiration. What's being oxidized, what's being reduced. Then Memorize the 4 steps of cellular respiration. Where do they occur? What comes out of them? Okay? And that's the way I approach this, because there is a lot to learn here. And a lot of times we learn something complicated and you're like, I'll never learn this again. Well, if you're having a hard time of why you think you should understand this, watch my earlier video on this, because I think it's really exciting and really important to learn. But another reason to learn this, even if you have no interest in cellular respiration, learning something complicated with lots of moving parts, lots of things happening, lots to memorize, lots to understand is important, because that's training your brain to learn things that might be difficult for you. It's like exercise, right? If you've never run a mile and you've been binge watching Netflix and eating popcorn all summer and somebody goes, hey, get up and run a mile, it's going to be hard. So the way you run a mile and eventually run further and even run a marathon is you practice a little bit every day, and your body gets better and better and better, and it gets easier to run as you continually work out. In fact, athletes work out and practice almost 2 hours a day for their sports. Learning is the same way. Hey, there's a lot that happens here with cellular respiration. The way we learn it is the same way you would practice if you played a sport, whether you swam or ran or weightlifted or played football, baseball, softball. You practice every day, right? You can't sit down and learn it all at once. Football players don't learn their season by cramming for a football game on a Friday afternoon. Okay, so learning is the same way. So even if you have, you're just like, oh God, I got to just memorize this to get through my intro biology course. I want to go learn about birds, which is what I was when I was an undergraduate, right? I wanted to learn about birds. I sat down and memorized it. And now as a teacher, I realize that there is a value in learning things that are complicated, because it trains your brain like you train your muscles. Okay, enough of that. Let's dive in here. Let's go. All right. Glycolysis. Oh yeah, concepts 9.2 and all this. This is just because I'm following the textbook to help my students know what sections to read. I'll probably have to redo this video when they come out with a new edition of the textbook. But here we go. Step 1 of cellular respiration is glycolysis. Now, importantly, glycolysis. Glyco has to do with glucose. Lyse means to split apart. So glycolysis, the name of it is telling you something. We are going to split apart glucose, okay? Now, glycolysis, we're going to start to oxidize it, right? And basically, it's going to occur in the cytoplasm of the cell. So even though I'm showing a mitochondria here, guess what? Glycolysis occurs in the cytoplasm outside the mitochondria, right? And there's some really cool things about glycolysis before I even get into it. It's an ancient metabolic pathway. Every cell does it. If every cell does something, that tells you some important pieces of information. 1, it tells you it's important. 2, it tells you it's ancient. 3, it tells you that our last common ancestor did this, and we've inherited almost unchanged for 3.8 billion years. That's amazing. And another piece of evidence that we know it started out early, not just because it's shared by all life, doesn't require oxygen. See, when life evolved on this planet, there was no free oxygen in the atmosphere. Now there's oxygen as the element bound to rocks and in water and carbon dioxide and all that, but there was no free oxygen, that's O2. And it occurs in the cytoplasm of the cell. Doesn't need any special structures to do it. And it takes ATP to make ATP in glycolysis. There's an energy investment stage to get glycolysis started. And basically what you do is you phosphorylate it. And here's why. You ever started a fire, burned with some natural gas? You know that you have to have a spark or some flame to get your redox reaction going. In fact, even exergonic reactions, even though they release energy, you need an input of energy to break the bonds, right? That's our energy barrier. So in glycolysis, we use a couple of ATPs, and the ATPs uses a kinase to transfer a phosphate to the glucose molecules, and you phosphorylate it. You create an active intermediate, right? And by adding phosphate to our glucose molecule, we add a lot of potential energy. And by adding potential energy to it, the bonds are easier to break. So with glucose, yeah, that's my rooster going out there. Okay, To get glucose started, we have an energy investment phase, okay? That's our phosphate groups. And then it goes through a series of reactions. We're not going to memorize them here. And then we're going to have an energy payoff phase. So glycolysis in the energy payoff phase, what's going to happen is we're going to take some of these electrons and we're going to pull them away from the glucose molecule, right? We're going to actually rip them off of the hydrogens and the carbons, and we're going to transfer them to NAD+, and it's going to thereby be reduced to NADH. And the reason why we do this is because these electrons carry energy, right? That's really important. And this is part of the oxidation of glucose. Now, if anybody wonders what the electron carrier looks like, it's called nicotine adenine dinucleotide. And here's a picture of it. You can actually see the adenine in there. But this is the reduced form of that molecule. And this is one of our 2 electron shuttles used in cellular respiration. Now, glycolysis also makes at least 2 ATPs through a process called substrate level phosphorylation. Now that's 2 net, because remember, at the beginning of our glycolysis, we had an energy investment of 2 ATPs. So we make 4 total, but 4 minus 2 is 2, right? So now we've got 2 net. And this is called substrate level phosphorylation. Now what that means is, anytime you hear the word phosphorylation, okay, I'm going to phosphorylate something. I'm going to stick a phosphate group onto it. And substrate level, substrate, that tells you enzyme and a substrate. And your substrate, what's going to happen, the enzyme is going to facilitate this reaction of adding a phosphate onto ADP to make ATP. And the way it does that is it does what is called an energetic coupling. It couples an exergonic reaction, to break down a glucose, to an endergonic reaction, the phosphorylation of ADP, okay? So this is not exactly the most efficient way to make ATP, something that I will talk about at the end of this slideshow, or at the end of this video. Now, glycolysis is a metabolic pathway. It is a series of 10 chemical reactions, okay? And basically, it's also regulated by what is called feedback inhibition. What that means is, as the cell produces more and more ATP, if it doesn't use the ATP, then ATP will actually allosterically inhibit one of the early enzymes in glycolysis and slow the entire reaction down so you don't build up too much ATP inside the cell. Believe it or not, as much as we need ATP, too much is actually a bad thing. It's a really reactive molecule, so you don't want a whole bunch of it sitting around, and you don't want to waste your energy making it. And of course, here's our regulatory site for feedback inhibition, and ATP regulates the rate of glycolysis, okay? So pretty cool stuff there. Now, at the end of glycolysis, okay, we've got 2 ATP nets, we've got 2 NADHs, this is per molecule of glucose, and we're going to basically split glucose into 2 molecules called pyruvic acid, or pyruvate if it's missing that hydrogen on the carboxyl group, okay? And technically, I think a lot of times when you're in like cells, that hydrogen pops off and that oxygen becomes negatively charged. Now, if oxygen is present inside the cell, or at least enough of it, then pyruvate will get transported into the mitochondria. Now, that's actually still an area of study. I mean, I think they've almost got it figured out, the type of transport proteins that are pulling it into the mitochondria and how it knows to do that with oxygen present. But once there, we go to stage 2, which is pyruvate oxidation. The name tells you something. I'm going to take pyruvate and I'm going to oxidize it. It is also called pyruvate decarboxylation. I actually prefer that name. I know it sounds more technical, harder to say, but decarboxylation tells you exactly what's going to happen. Now, you guys remember your functional groups, right? Hydroxyl groups, carbonyl groups. Don't forget that carboxyl group. Look at the top of that pyruvate. See? The double bond to an oxygen and an O minus. Decarboxylation, that's a carboxyl group. D means without, right? So if we have pyruvate decarboxylation, that carboxyl group is going to come off and form carbon dioxide. So each pyruvate is going to lose a carbon dioxide molecule. So pyruvate oxidation, well, we're oxidizing our pyruvate molecules. Redox reactions. Something got reduced, something gets oxidized. In this case, our electron carrier, NAD, is going to get reduced to NADH. And then we've got a 2-carbon compound, acetyl, and it will be attached to a coenzyme A and form acetyl coenzyme A. And then coenzyme A is a pretty cool molecule, right? Let's see if we can get a picture of it here. There it is. It doesn't fit neatly into one of the 4 macromolecules. Coenzyme A is this really fascinating molecule. I mean, you can see that it's got a nucleotide in there, some amino acids, and it binds to that acetyl group there at the very end. And what this molecule does is it links pyruvate oxidation to the Krebs cycle. So it's bringing in these carbons that ultimately started out in glucose. Now, here's where we come into the Krebs cycle. This is also called the citric acid cycle because it goes round and round and round and it makes citric acid. It took Hans Krebs nearly a decade of his life to figure out the chemical steps involved with this, and he actually earned a Nobel Prize for his work. And interestingly, the citric acid cycle, like glycolysis, occurs in most living things. Even though it occurs inside the mitochondria in eukaryotes, many prokaryotes carry out the Krebs cycle. And the general purpose of the Krebs cycle is, here's a more detailed showing of all the carbons in it and what happens to them. But basically, we're going to fully oxidize our carbon, right? We're going to rip the rest of those high energy electrons off and the hydrogens, and we're going to stick them onto our electron carriers. So for every molecule of glucose that we started with, that created 2 pyruvates, which got oxidized to 2 acetyl coenzyme A's, right? And they're going to come into the Krebs cycle. And then for each one of those acetyl coenzyme A, we're going to get 2 carbons. That'll make a molecule of ATP. It'll make a molecule of FADH2, which is our other electron carrier. And then it will also make 3 NADHs. So for a total of 2 ATPs, 2 FADH2s, and 6 NADHs. And by the end of the Krebs cycle, all of the carbon has been oxidized to carbon dioxide. And so our glucose molecules they're oxidized. They're gone. Now, a couple things here. We haven't seen water, right? We haven't seen oxygen. Now, interestingly, the Krebs cycle is also regulated by ATP. So if you start getting a lot of ATP, it's regulated by feedback inhibition. And also, interestingly, the Krebs cycle can run backwards in some prokaryotes. If you add ATP to it, you can actually fix carbon dioxide. And it's like one of 4 metabolic processes that we know of that can actually fix carbon dioxide. Pretty wild, isn't it? But that's not what we're going to talk about here. But just keep in mind, we haven't talked about oxygen. We haven't talked about water. So here we go. We went through glycolysis. We went through pyruvate oxidation. We went through the Krebs cycle. And what have we done? We've oxidized our organic molecule. We have ripped off all those high energy electrons and hydrogens. We have transferred them to our electron carriers, NADH, FADH2. And in the process, we have made a total of 4 ATPs. Yeah, not very much, isn't it? So here's where the magic starts to happen, right? And we can see that in our free energy reactions as we do this, it keeps going down, down, down. Now, cellular respiration is incredibly efficient for extracting energy. It's about 40% efficient. About 40% of the energy stored in glucose molecules gets transferred to ATP. And of course, you know from the laws of thermodynamics that the rest of it is generated as heat and is lost because of entropy. So how do we make all this ATP? It sure can't be substrate level phosphorylation. Here's where the magic happens. Oxidative phosphorylation. This to me is a secret to life, right? Or at least chemiosmosis. Now, oxidative phosphorylation, this requires the inner membrane of the mitochondria. This is why mitochondria have a double membrane with all of those folds inside of it. And that is important because those folds increase surface area for oxidative phosphorylation to occur. Now, oxidative phosphorylation, once again, the name is telling you something. We're oxidizing something and there's some phosphorylation going on. Remember, phosphorylation means I'm sticking phosphorus groups onto something. In this case, ADP to make ATP. The oxidation, of course, is our electron carriers, right? We've already oxidized carbon in our organic molecules. They're done. They're out. They're diffusing out of the cell. But here we go. We're going to oxidize our NADH and FADH2 to power an electron transport chain. And then the electron transport chain, in turn, is going to power chemiosmosis. And like I said, chemiosmosis to me is fantastic. Even the electron transport chain is. And even though, I got to take a side note here. I got to go on a tangent. We present this as though we know exactly what's happening here. That is not the case. We know a lot of how it works, but it turns out that quantum effects of tunneling and wave functions and decoherence are important for this. So yeah, it's more than just classical physics. There's some quantum effects that may be affecting how this works. And like I said, how the electron transport chain works and these membranes and how the electrons get transported between these electron transport complexes is an area of active research. I mean, we talk about these electron carriers in the membrane, but the reality is the electrons are probably moving in waves, and then when they get to the complexes, they stop acting as waves and act as particles, and then act as waves again. Isn't that weird? It's so awesome. Okay, so here's the electron transport chain, right? The electron transport chain is made up of 4 large molecules. Well, actually, it's made up of more than that. Most of them are these electron complexes, 1, 2, 3, and 4, with these smaller molecules that are shuttling the electrons between these transport complexes. And like I said, that movement of these electrons between these complexes is still an area of active research and probably involves some crazy quantum mechanics here. So you can see the close-up. This is occurring, of course, on the inner membrane. Now, big picture. FADH2, NADH, they're going to get oxidized. They're carrying energy from our organic molecules. And as they get oxidized, they're going to dump their electrons off on the electron transport chain. Specifically, complex number 1. So if you were going to go from left to right, complex number 1 is going to oxidize NADH. So it's going to rip off its electrons, and it's going to use those electrons to do work, specifically active transport. It's going to pump protons into the intermembrane space. We're pumping protons into the intermembrane space. And then complex 2, which is that smaller one, that oxidizes FADH2 to FAD. And then the electrons are going to get pulled down in energy as they go down the electron transport chain. And you can see the little red arrows. They are used to pump hydrogen into the intermembrane space. Okay, that's important, right? So now the energy that was stored in a molecule of glucose or whatever organic molecule we oxidized, some of that energy has now been transferred to an electrochemical gradient, okay? And this electrochemical gradient, it is really important for chemiosmosis. Chemi means chemical, osmosis means to push. All right, so, I keep saying this thing is a secret to life. I hope I'm using the word the way it should be used. But you see that other molecule down there? It's called ATP synthase. Okay, ATP synthase. This is an enzyme. ACE, A-S-E, tells you it's an enzyme. Synth means put together. ATP synthase. I wonder what it could be doing, like making ATP or something. Okay, as we create this electrochemical gradient, we get an electrical gradient, right? Because we're pumping protons. Protons are positively charged. That means the intermembrane becomes positively charged compared to the matrix of the mitochondria, which becomes negatively charged. That's a voltage. That's storing potential energy just like a battery does. That's your electrical gradient. Because there are more protons on one side of a membrane than another, that is a chemical gradient. Hence, you put them together, you've got an electrochemical gradient. And these things are out of equilibrium with the matrix, and they want to be in equilibrium. The universe is always trying to be in equilibrium because it's stable that way. But the protons are ions. They can't cross the membrane very easily, so they flow through the ATP synthase. In fact, they're like pushing through. Just think of water behind a dam, right? As water gets stockpiled behind the dam, it pushes its way through. That's chemiosmosis, right? Pushing. So as it pushes its way through the ATP synthase, sounds like facilitated diffusion here, the cell can harness that to do work. And what it does is it does work by using that kinetic energy of those protons to make ATP. Okay? Interesting, huh? And it makes a lot. And it's called oxidative phosphorylation, once again, because we're oxidizing our electron carriers, NADH and FADH2, to phosphorylate ADP to ATP, doing chemiosmosis, all right? And this is important. I mean, every single cell in this planet makes ATP using chemiosmosis across their membranes. Prokaryotes do it. We do it in our mitochondria. In fact, because we can pack so many mitochondria in our cells, they can become much larger, and we can actually have multicellular life because we can make so much ATP. And not every cell uses oxygen as an electron acceptor. Some have to use other elements that involve nitrogen as an electron acceptor, and they don't make as much ATP. And in fact, because prokaryotes are making the majority of their ATP across their membrane, that limits them in size, right? And that is important. Now, the fact that cells like ours use oxygen, that profound consequences for the future of life, right? So one thing I haven't talked about, oxygen. Where do the oxygen and water, where do they come into play? And you need to know a little bit about oxygen, right? It's electronegative. It loves electrons, right? And if we got an electron transport chain, and those electrons are going down the transport chain, you have to remove them. And like I said, other prokaryotes, there are some prokaryotes that don't use oxygen. They use nitrogen compounds as their electron acceptor. Nitrogen is more electronegative than carbon or hydrogen. But when you use oxygen, what happens is those electrons can go way down in energy, which means you can create a larger electrochemical gradient. You can store more energy. It's like storing more water behind the dam, right? And by doing so, by getting more work out of those electrons, going all the way back to your organic molecule, right? By getting more work out of them, you make more ATP. This tiny evolutionary step. And if you look on the right, you can see 1, 2, 3, 4 complexes. The bottom right would be complex 4, and that is cytochrome oxidase, right? Cytochrome oxidase takes the electrons and the oxygen, lines them up, and puts the electrons on the oxygen, which is an easy thing to do. And we get a lot more work out of our electrons, we get way more ATP. But by using oxygen, and then not only does it just put electrons on oxygen, that would just create a ROS, free radical, right? Reactive oxygen species. Not only does that, it takes the protons and puts them on the oxygen and forms water. Now all of a sudden you've got this molecule, this in your cells oxidizing everything. Let's put you to work. You put it to work, you get a lot more ATP, and then in the end of it you make water. And of course water is basically a requirement of life. It's harmless for life for the most part. Wow. Isn't that amazing? That one little evolutionary innovation of sticking electrons on oxygen changed the world. And it paved the way for multicellular life. I mean, that's why it's so important. So let me go back, and I want to talk about more of why chemiosmosis and the electron transport chain are so cool. Here's another reason why. Earlier, in glycolysis and in Krebs cycle, you make ATP through a process called substrate level phosphorylation. And in fact, up until the 1960s, most scientists believed that the majority of ATP made in mitochondria is made by using an enzyme in substrate level phosphorylation. And remember this, to do substrate level phosphorylation, you have to couple an exergonic reaction with an endergonic reaction. Now, it takes 7.3 kilocalories a mole of energy to make ATP. Now, if you've got 7.29, too bad, no ATP. If you have, 30 kilocalories a mole of energy from your exergonic reaction, great, you get ATP. You don't get more ATP, you just get one. That's what happens when you couple an exergonic reaction with an endergonic reaction. That's that energy coupling and substrate level phosphorylation. I know a lot of words, right? Here's another way to think about this. I don't know about you, but I like my Mountain Dew. I drink diet these days. And basically, let's say you want to go to a vending machine. It takes $1.50 to buy a Mountain Dew. It takes 7.3 kilocalories a mole of energy to get an ATP. Okay? If you get $1.45 and you put it in the vendor, you don't get 90% of a Mountain Dew. You get none. And the machine keeps your money. Sucks. Energy coupling is the same way with substrate level phosphorylation. If you're trying to use an enzyme to make ATP, right, by getting the energy from another exergonic reaction, and you don't have enough energy, you lose that energy. It's gone. It's gone as heat. All right, now, let's think of it this way. Let's say you get a $5 bill. You stick it in the Mountain Dew machine. You pop out a Mountain Dew. Fantastic. Machine keeps you $3.50. Yeah, that's bad, right? You get no change back. If your exergonic reaction produces a lot more energy than is needed for an ATP molecule, then that extra energy is lost. You get your ATP, but you don't get change. This is not efficient and it's wasteful. So oxidative phosphorylation, it solves this problem. Okay, so here's what happens. We got these first 3 stages of cellular respiration, right? We're oxidizing glucose. We're ripping off those electrons. We're ripping off the hydrogens, and they each have energy, right? I mean, we make a little ATP along the way, but we're taking that energy, we're transferring over to the electron transport chain, and we're going to store it. We're going to store it as an electrochemical gradient. It's like taking your change and putting it in your desk drawer, and then when you have $1.50, you go buy a Mountain Dew. By doing all these little steps of the reactions, we're taking that energy in small pieces, storing that energy as an electrochemical gradient. We're storing that energy in the intermembrane space. And when we have enough to make ATP, we make ATP. And we waste much less energy. This is brilliant. I mean, it makes 10 to 15 times the amount of ATP, right? Isn't that awesome? And I have to talk about the history of this. This to me is pretty exciting here. Okay, in the 1940s and 1950s, they were trying to figure out how mitochondria made so much ATP. Then this scientist, Dr. Pete Mitchell, he said, it's chemiosmosis. And everybody looked at him and goes, man, that is so ridiculous. No way. But he proposed it, and he said, this is how they're making it. And he realized that the electrochemical gradients inside the mitochondria was enough to make ATP. And he was vindicated when they discovered ATP synthase a few years later. Guess what? 1978, he wins a Nobel Prize for his work. And at the time, he was considered a maverick scientist because his ideas were so revolutionary that they were so far out there, people didn't believe him. He was right. Now, of course, for all you 80s film fans of Top Gun, one of the most awesome movies ever, starring Tom Cruise as Lieutenant Pete Mitchell, Maverick. I love the fact that Pete Mitchell is the name of an awesome scientist and a cool character in one of the greatest 80s films of all time. It's infinitely quotable. And of course, it gave us a great song, the awesome song, Highway to the Danger Zone by Kenny Loggins. And that song is so good that Archer kept bringing it up throughout season 5. And then at the end of season 5, guest appearance with Kenny Loggins. Okay, well, there we go. I hope you enjoyed this overview of cellular respiration, and I had to end on a little bit of pop culture there at the end. Okay, well, last thing here. I used a lot of words. Substrate level phosphorylation, oxidative phosphorylation, energy coupling, exergonic reactions, endergonic reactions. You got to slow down on this video. You got to take notes. You got to memorize these foundational terms, right? And when you memorize them and I start ripping through that, it will help you understand it so much better. To memorize those words before you watch the video and then memorize them and watch videos like this again. And like I said, this is another video on cellular respiration. And this is Dr. Tom Kennedy. Until next time, we'll do fermentation.